So I'm going to talk about the role of genetics in a child with liver disease. So this is the outline of my presentation. So first, I will give a brief introduction, how the importance of genetic platforms in terms of liver disease and classification and types of genetic testing which are available for liver disease screening, and also the importance of genetic testing in liver diseases and what are the available genetic testing in Sri Lanka. And finally, we will summarize the genetic testing so far we have done in CNCLD clinic. So let me begin with the introduction. So over the last 30 years, genetics has enhanced our understanding and management of liver disease. With the advancement of genetic platforms, the genetic testing has become a kind of like mandatory test in every medicine, every discipline of medicine. So also, if you take the immediate future, the genetics will allow further stratification of liver disease, and it will definitely contribute to the personalized medicine and customized treatment. So if you take the monogenic liver diseases, so nearly half of the chronic liver disorders presenting in childhood, they have a genetic basis. Roughly about like 20% of liver transplantation in children, they are performed as a consequence of liver-based monogenic diseases. And also most chronic progressive liver diseases, except biliary atresia and autoimmune liver diseases, they are inherited liver disease. So this classification, the childhood liver disease, we can classify into three categories depending on the genetic presentation. And it is kind of like our classification. So we can divide into patients with cholestasis, so like Alagille syndrome, PFIC types, citrulline deficiency, and also the patients with elevated liver enzymes, mainly the Wilson disease comes into this category, and also congenital disorders of glycogen, and also the patients with hepatomegaly or hepatosplenomegaly, like mainly the glycogen storage disorders, and apart from that, Niemann-Pick disease and Gaucher disease. So if you take genetic testing in general, there are three types, cytogenetic testing, molecular genetic testing, and molecular cytogenetic testing and molecular genetic testing. So the cytogenetic testing, mainly we concentrate on chromosomes. So it is that's not important in terms of liver disease. So the molecular cytogenetics, mainly we are dealing with fluorescent in situ hybridization and array. You can detect kind of like small deletions and duplications. And the molecular genetic testing, mainly we are dealing with DNA and RNA. So in terms of liver diseases, the molecular genetic testing platform is the platform in genetic investigations. So if you take the commonly available molecular genetic testing techniques in everywhere in the world, you can divide into mainly two types. So the Sanger sequencing-based platforms and the next generation sequencing platform. So the Sanger, you sequence one gene at a time, and it's very time consuming and it is not practical. So nowadays we hardly use this unless you have a validation process, or if you know the family mutation. For the carrier screening, we use Sanger. So the next generation platforms, sequencing platforms, are the investigation of choice nowadays. So there are different types of testing under this category. So targeted gene panels, that is, we take limited number of genes. Could be a liver panel, could be a cardiac panel, could be a carrier screening panel, like that. It could be a cancer screening panel. We are targeting on a particular disease group and select those genes and try to compare the available mutations with the patient mutation. And it is low cost, but you can't use this technique when the phenotype is uncertain, and we will be in trouble. And the clinical exome sequencing, like if you take the genetic disorders, roughly about like 4,000 genes have been discovered as genetic diseases, genes that causes genetic diseases. We take the exons of all these genes and then we sequence in one go. So this is also kind of like practical, but then again, you can't detect novel mutations. And also if the phenotype is uncertain, you can't use it. If you take whole exome sequencing and whole genome, you know, the word per se, the whole genome is the whole human genome you sequence in one go. So both in exons, introns, the regulatory regions, everything we sequence, and it is not actually practical. So in research basis, so maybe if it is really, really indicated, otherwise we don't offer this whole genome sequencing because it is pretty expensive. Whole exome testing, it's kind of has become the testing modality in lot of medicinal, lot of disciplines of medicine. So whole exome, you take all the coding regions, you know, if you take the human genome, so about like 3% of the human genome is the coding sequence. So you take that genes together and do the sequencing in one go, and then you decide whether there are mutations or variants which can be compatible with the patient's clinical condition. So the most important thing in this whole exome sequencing is, if you know the phenotype, you can offer this testing. Also, with in silico analysis, with computational tools, we can detect novel mutations. If the clinical condition is compatible with that, we can confirm it. However, we have to confirm by doing finally the protein studies. So in next few slides, I'm going to talk about the importances of genetic testing in pediatric liver disease. So I'm going to elaborate these importances with our clinical experience, with our clinical details. So obviously, the genetic testing, it helps to diagnose and confirm the clinical condition. So if you take this case, she's a 7-year-old girl with a family history of neuro-Wilson disease in her brother. So she was the proband. She came to the CNCLD clinic, and the brother was diagnosed to be having neuro-Wilson disease. And this child had slightly high ceruloplasmin level, slightly low ceruloplasmin level with 24-hour copper was slightly high. And liver functions were all throughout the time, it was normal, and genetics were not done in this family. However, based on these investigations, this child has put on penicillamine and zinc. So then our team, of course, they had a doubt, you know, even for Wilson's carriers, there could be like slightly low ceruloplasmin level with slightly high 24-hour copper. So we wanted to do the genetic studies in this family. So first, what we have to do is, if you do a genetic testing, you have to do the affected patient in first. And then depending on we had to find the family mutation, and then we can go for carrier screening. So what we did was we did the genetics testing on this brother, and then we were able to find the genetic mutations of the family. And finally, we did the carriers, the same mutations we analyzed on this baby girl. And it was found to be having a Wilson disease carrier. So the message what we can get from this case is, you know, like within certain period, within few months, with the genetic testing, the child, being a patient of this child, became a kind of like normal carrier. So it is very important. So we reassured the patient and chelators were stopped. If you take this case, so she's a 1-year-old girl and presented with persistent jaundice. So she's the proband, presented with persistent jaundice, pale stools, dark urine, and then ultrasound abdomen, it was detected to be having large hepatomegaly. So if you take the pedigree, it was noted there were 2 sibling deaths, and it was like with the clinical notes, it was confirmed as congenital cytomegalovirus because the mother had CMV IgG positive. So when first case was labeled as congenital CMV, of course, the CNCLD team accepted it. But when it comes to another second case that was reported, so then the diagnosis was doubted. So then we did the CNV IgG IgM antibodies of this child, and it became negative. So finally, the team decided to do the genetic testing, and it was found to be having PF5C2. So then, luckily, the child didn't label as CMV, and then the manage accordingly according to the PF5C management. If you take the third case, again, she's a 13-year-old girl, was treated as an autoimmune liver disease, and the steroid was initiated, and steroid toxicity was reported, and the response was poor. And the liver functions were high, and genetics was not done. But however, since the diagnosis was doubtful, CNCLD team, they did the whole exome sequencing, and finally, we were able to find that it was a Wilson disease case. So all these three cases, you can identify that the diagnosis was completely changed with genetic testing. And the other importance is it helps in the family screening and early diagnosis in siblings. So this one is a 12-year-old boy presented with clinically diagnosed Wilson disease and with compensated chronic liver cell disease. The child was awaiting liver transplant. So however, since the child was awaiting liver transplant and the child had two other siblings, the CNCLD team decided to go for genetic diagnosis of this child. And then we found there were two mutations in the ATP7B gene. One was a reported variant, the other one was a novel variant. So this is, I mentioned you earlier, the advantage of doing whole exome sequencing. So the one mutation was reported and it was there, but then again with in silico analysis, we were able to find another mutation. So this is a compound heterozygous case where the two alleles were different alleles when it comes to Wilson disease cause. And then we did the carrier screening. We did the Sanger sequencing of the other two siblings. And I told you the Sanger sequencing is important when it comes to validation process. And also, if the family mutation is known to you, you can do the Sanger sequencing of the other siblings. So unfortunately, we found that this 1.5 years old child, and this child didn't have any symptoms, and the child was found to be having Wilson disease. And then, so the CNCLD team initiated the chelator treatment. And the next importance is the next pregnancy planning and counseling. So, you know, we are dealing with genetic disorders. Invariably, there is a certain percentage of recurrence. So the next pregnancy planning and counseling is mandatory. So, so I'm going to elaborate that importance with this case. This one is an 8-year-old boy from a consanguineous marriage and was found to have high AST level. And clinically, child was asymptomatic. But there was large hepatomegaly was noted, and the child has had 2 episodes of hypoglycemia with 6-hour fasting. So the clinically and histologically, it was proven to be having glycogen storage disorder. But however, this couple, they were willing to have another child. So then we want the genetic diagnosis before planning for a next pregnancy. So we did the genetic confirmation, and it was found to be having glycogen storage type 6. So then we got down the family and we did the reproductive counseling for the prevention of recurrences. So we discussed all possible reproductive options which are available in our settings, starting from prenatal testing. You can do amniocentesis at 16 weeks or chorecentesis at 18 weeks of gestation. And we can, because the family mutation is known to you, we can do the genetic testing, whether the baby has affected, to see whether the baby has affected or not. And also we offer them the pre-implantation genetic testing coupled with in vitro fertilization. So this is also available for if the family mutation is known to you, we can test the embryos before being implanted. And also embryo donations. So autosomal, since this condition was autosomal recessive, you can go for donor eggs, donor sperms. And if if any of those options are not possible with the couple, so we can offer them the adoption. So depending on their choice, we only can give them the options. Depending on their choice, we will assist them for future pregnancy to prevent the future recurrences. So the other importance is, like we are in the era of personalized treatment. So if you know the phenotype-genotype correlation, so you can predict the disease nature, and also based on the mutations, you can plan the management. You can personalize the treatment. That is also very important. And also the cost effectiveness. So most of you people might think that it is not cost effective for doing genetic testing, because I agree that most of the genetic platforms, genetic testing investigations are pretty expensive. But if you take those advantages I mentioned you earlier, if you take those clinical cases I mentioned you earlier, and also, some patients, they are coming to clinic more than 10 years without a proper diagnosis. They have like multiple clinical books without a proper diagnosis. And they are considering their costs, their transport cost, everything. So we believe that doing, genetic testing is doing only once in lifetime. Doing once in lifetime a genetic testing, we believe that it is cost effective. So these are the available genetic testing in Sri Lanka. Okay. So in terms of liver diseases, we have molecular genetic testing. We have considering the molecular genetic testing, we have Sanger sequencing, especially for validation or maybe carrier screening. And then we have NGS-based platforms. We have the whole genome and whole exome. Of those the NGS platforms, I must mention this whole exome sequencing. It's very important testing. It has become a kind of like the testing of CNCD liver team now, because now after having a series of discussion with one of the private laboratories in Sri Lanka, so we were able to acquire a kind of a like very concession price for whole exome sequencing. It's remarkably low. And it's a non-profit basis service. And along with the genetic testing, with the genetic report, we do a multidisciplinary counseling, including myself, genetic counseling, and also Meranti, we do a multidisciplinary counseling of the patient, and we discuss the carrier screening, the next pregnancy counseling, and possible reproductive options and their management. Everything will be discussed. So if you do have any sort of like patient with uncertain diagnosis, if you do want to do a genetic testing, you are free to contact the pediatric liver demonstrator. At the moment, we have Dilshan. So we can contact this person. We can divert your samples through this channel, because it is remarkably low when you compare the whole exome sequencing prices in other countries. And finally, I'm going to share our genetic analysis results. So far, we have done in the CLCD clinic. So mainly, we have done the whole exome sequencing. So we have done about like 60 odd samples so far. And of those samples, there were about like 14 Wilsons. And there were 4 PFICs and 6 allergy syndrome, 6 glycogen storage disorders, and about like 18 other patients for 1 each, and some other genetic causes for 1 each, like, Niemann-Pick, Dubin-Johnson, galactosemia, congenital disorder of glycosylation, and Joubert syndrome, Coffin series, like that. There were like 18 other conditions, and also in conclusive reports with non-genetic, including 9 patients. And I think that's what I have to tell you. And these are my references. And thank you so much for listening to me. If you do have questions, I can answer at this moment.